Hi everyone, good morning. Today we met to discuss continuation of our lesson reproduction. So we have discussed in earlier classes asexual reproduction. We have studied some only from this topic. You know which are they? They are binary fission in amoeba, then multiple fission, then we have studied budding in hydra and yeast. You know the differences of budding in yeast and then hydra. So from the buds are developed. The buds are developed from in the hydra, from the lateral sides of the body. But in the case of the yeast is a unicellular fungus. Then the buds are developed from the top part of its body. Then they forms a chain. Then it is detached from their parents and develops into an adult. So here they multiply in number. So in this we are going to discuss spore formation. What is spore formation? Which organism exhibit spore formation? Especially you also studied about bread mold. What is its scientific name? That is Rhizopus. Rhizopus, penicillin, then mucor. These are some of the fungus they exhibit spore formation. That means they multiply in number. They are asexually reproduces by the process known as spore formation. So in this case, in this process, spore formation, spores are produced inside of their body. That means they are specially produced as structures, a cup-shaped structures or blob-like structures on their top part. It's a structure. For example, we have taken as an example here bread mold or rhizopus. So you can see a fungus, it bears, it has the stem-like portion that we called it as hypha. Then it has a rough portion that called as a substratum. What we called a substratum. It is attached on this surface. That is a substratum. It is not. It doesn't have, all the fungus doesn't have what a true roots and stem. But we called it as stem is called as a hypha. Then its root, that is rhizoids. In any case, they have the rough surface. They have something false roots. They have. It is used for attaching on a rough surface. We called it as substratum. Okay. So then they have a specially adapted structures to bear the spores. They are called as sporangium. What we called the structure is called as sporangium. But to draw the diagrams, So this is the substratum. It means a rough place that is called as substratum. This is the hypha. Hypha. If it is plural, we can write hyphae. So hypha. Then it has a specially adapted cup-shaped structures. This structure is called as a sporangium. The sporangium encloses spores. What? Spores are enclosed in sporangia. Then in all the process asexual reproduction we have studied, at first takes place karyokinesis. What is karyokinesis? That is the division of nucleus. Then it is followed by cytokinesis. You have studied well. All these process are taking place here is also in the case of the spore formation. The spores are produced here. Number of spores are produced. So during favorable condition comes, so what happens? It breaks this sporangia, this cyst, and they are releasing a number of spores. So this each spores develops into an adult individual. That means they grows, each one grows into whatever they are adult individuals. Are you clear? So it's very important and study the diagram also. So once again listen. So they have the structure that is called as sporangia. Inside of the sporangia it encloses spores during favorable condition. So in this case, when it keeps these spores inside of the sporangia, they are taking place karyokinesis at first and followed by cytokinesis. So we have studied a little bit of cytoplasm around the divided nucleus. You know that we have studied. If you studied well before taking part, it is very easy, I think. Okay. So at first you study the before taking portions and then only you can understand that. So then the sporangia consisting of spores during favorable condition comes, it bursts, desist and also liberated number of spores. Each spores develops into whatever. Yes, it's an adult, grows into an adult. Are you clear? So favorable condition means here they need moist surface. So if that is favorable for them, moist content is there, then that is its favorable condition. So spore formation occurs in fungus and bacteria also. Okay? Yes, it's very important. So the next, so this is process is called as spore formation. That is noted in rhizospores. Study examples. Then another example that I already said to you, that is mucus, and plus penicillin. So these are fungus. So next, the process is called as fragmentation. Then the third process, that is called as regeneration. So first we are going to discuss fragmentation. You know what is fragmentation? So we want to discuss what are the differences between fragmentation and regeneration. It's very important. So you know what is fragmentation? Fragments means a piece. A piece is produced from the matured cell. That is fragments. You know that from where it is developed, which organism exhibit this process, that is Spirogyra. You know that which organism is Spirogyra, where its habitat, that is we called it as, it has a nickname, that is pond silk. It's familiar for you, I know it well. So then fragmentation means from this organism a fragments or a piece is produced. So each fragments they are develops into or grows into another Spirogyra. So Spirogyra is a multicellular organism. You know that what is multicellular organism? They consist in number of cells are more than one cells. They are called as multicellular organism. In this organism, each fragments they have the capacity or capable of breaking their cells. These cells are develops into or grows into an adult individual. How? So at first I told you, it is a process, it's a multicellular organism. So relatively it has a simple body designs. That is the most important in this case of Spirogyra. Because it is a multicellular organism but it possesses a simple body designs. Then it reproduces by cell, that is a simple divisions. It still works here in the case of Spirogyra. But in the case of the multicellular organisms, we have studied that cell to cell division is not possible, that is impossible. Why? Because the reason is that cell is divided at first and then forms tissues. Tissues again organized together to form organ, then organ systems and a whole organism. So what happens in the case of the Spirogyra, it is impractical in multicellular organism, but it is only possible where in this organism it reproduces asexually because of its body designs. It possesses simple body designs. Are you clear? Yes, which organism exhibit fragmentation, that is spirogyra. Repeat. So next is regeneration. Some of the organisms such as hydra, then sponges, then planaria. These are the organisms, they exhibit this process, asexual reproduction method, but that is regeneration. What is regeneration? This is also, we want to explain that regeneration, it is the capacity of the organism to replace its lost body parts. You understand? To replace its lost body parts. For example, in the case of the planaria is the organism, it exhibits more regenerating capacity than hydra and sponges. You know what is the reason? So we have studied in which phyla planaria belongs, that is belongs to Platyhelminthes. It is a free living organism. I don't discuss this because we have studied that. And sponges, that is the belongs to the phylum Poriferous. Sponges also noted this process. You know, and hydra exhibits budding. So here is some specialized exceptions are there, because in the case of the planaria, you know, so planaria, the structure of planaria, you can see that. So this is planaria. So if the head lost, the organism lost its body, that is head part, which can replaces its head. If it loses the middle portions or tail portion, it also replaces its lost body parts. So now, which organism exhibited more regenerating capacity, that is planaria. You know what is regeneration? Regeneration, it is a process, it is one of the asexual reproduction process by which the organism can replace its lost body parts. So children, find out the differences between fragmentation and regeneration. In each case, the forms, each piece or fragments are developed, but their lost body parts can replace us. Then think and how. You can write it on your notebook. What is differences between fragmentation and regeneration? That I told you. Now, so the differences you have to write that. Okay. So the next another method, that is called as vegetative propagation. Vegetative propagation. What is vegetative propagation? So we have studied many methods of reproduction, that is sexual reproduction, how an organism can multiply in number. So we have followed many ways. So the last one is vegetative propagation. What is vegetative propagation? We know the vegetative parts of the plants. Which are the vegetative parts of a plant? Stem, root, leaves, etc. You know? You also know that you have studied that the edible part of a plant, that is stem, then you know roots, then fruits, then seeds, etc. Flower also, it's an edible part. So here vegetative parts, how an organism reproduces by this method, vegetative propagation. What is the definition you want to give for vegetative propagation? What is vegetative propagation? A plant develops not Y seeds from their parts, vegetative parts such as root, stem and leaves. Which are stem, okay, root and leaves. So these are the vegetative parts of the plant. So we have to study it. So by a stem, so we can propagate many plants by a process, by cutting process, you know that stem cutting, through stem cutting. You know, find out examples, you know, rose, then tapioca. Many examples you can find out from your surroundings. Then from the root, you know that, curry leaves, roots of curry leaves from the red and goa is another example. Why? Through seeds also it can develop, but so from their roots new plantlings are developed. Then leaves, you know that leaves, that is bryophyllum. Have you seen bryophyllum? Then epiphyllum. So So in Malayalam we call it as cl11. Epiphyllum is called as Nishagandhi. These are the two plants. They are produced, so that is, they can develop new plants by a process known as vegetative propagation. That is from their leaves. So you can see that. So their leaves. So this is the leaves. So here this is called as leaf notches. From where the roots are developed from their notches. So this is called as leaf margin. In their leaf margins you can see notches here. So from each notches they start to develop their roots. You can see clearly. If you will get the leaves of bryophyllum, you have to do that because you can keep this in the soil or in the pot, and keeping it is horizontally, not vertical, horizontally. So then start to develop. Roots are developed from each noses. From each nose, you know that they again start to tender leaves. Tender leaves arise from their part after producing their root. Okay, so then it develops into what? New individuals or new plants. Are you clear? So that is noted well in the case of what? Bryophyllum and epiphyllum. So study examples: epiphyllum, bryophyllum. So from their leaves they are arranged. New plants are developed from the leaves. So other new plants are developed from the roots. Which plants? From the roots they are produced from their roots. Then stem. There are three methods artificially for this process, that is vegetative propagations. It's a commonly, that it is first layering process, cutting process and grafting method. I think it is familiar for you. Grafting, then cutting and layering process. So by a vegetative propagation it has many advantages, that. Now you think about it. What are the advantages of vegetative propagations? Why we are introducing this process? So which plants? So it is applicable to which plants? You know that not many plants they are produced seeds. Some of them they lack of to produce seeds. So these are applicable in such a plants. For example, banana, orange, etc. They are not produced by seeds. So because they have false seeds, from these doesn't produce water plants. So this method is applicable to such a plants. They are not produced seeds. Such a plants cannot produce seeds. Through the seeds we cannot develop the plants. So it is applicable. Then also another thing is that so all the plants are produced. We have produced many plants, newly plants. So these newly plants are genetically similar because they are produced from the mature parent. That is why all genetical characters are easily transmitted, and also we can say that all the plants are genetically similar to their parents. Are you clear? So then third importance or advantages of applying vegetative propagation is that we can produce seeds, flowers and fruits easily than we produce the plants through seeds. Why? Natural. Because that they have limited periods. So if a plants we are cultivating a plants, it takes more time to produce flowers and seeds, etc. Because the plants are directly produced from their parents, that is why it can easily produce fruits and flowers. Whatever it may be, that is whatever there is from them, we get it easily. So these are the advantages of vegetative propagation. Okay. So that's all about asexual reproduction. Once again, different methods we have studied asexual reproduction. So in the last class we have studied binary fission that occurs in amoeba. It's very, very important, all asexual reproduction methods. Study very well. So then binary fission, multiple fission. So which of the organisms exhibit this process? you study all the diagrams. Study to draw all the diagrams. Everything you must study. So, the next binary fission and multiple fission, budding, two types of budding. Then, next we have studied about spore formation. Then now we have studied fragmentation, regeneration and vegetative propagation. So that's all about asexual reproduction. So write down some questions. Draw diagrams and explain the following. First, spore formation, then fragmentation, regeneration. Then next question, what is vegetative propagation? Write examples. Then the third question, what are the advantages of vegetative propagation? So now we have discussed asexual reproduction methods. Then on next class we will take sexual reproduction. Okay, so I can wind up.